as calcium researchers that calcium is going to be key in the regulation of some of those pathways. And likewise, as calcium researchers, we know things like spatial localization of the calcium signal, amplitude of the calcium signal, or the frequency of calcium oscillations and the nature of calcium changes is really how things like cell death, gene transcription, proliferation, and cell migration actually occur. So it's not surprising that really there is going to be this overlap now between calcium signaling and pathways that are important in cancer progression. So we're very lucky in the calcium signaling field that compared to other cancer researchers that we have a nice suite of pharmacological modulators that we can use in our studies and exploit. And that's not the case for often a lot of targets in cancer. There aren't yet drugs for us to work on, but we're very lucky in calcium signaling because if we just look at calcium influx pathways, for example, and ways that cells might increase their cytosolic calcium levels, we've got pathways like the ORA1 store-operated calcium entry pathway, and we already have really nice inhibitors, pharmacological inhibitors like Sinta66 for that pathway. Likewise, we have voltage-gated calcium channels, and we're very lucky again to be able to have even clinically used drugs which are able to inhibit those calcium channels. And when we look at the Nobel Prize from last year, even if you look at those more recent calcium channels like piezo-1, the force-activated channel, we have activators like YOTA-1. And then we look at the TRP channels, we see temperature-regulated channels like TRPM8, having natural ligands like menthol. And also we have drug development or pharmaceutical developed drugs that target TRPV4, like this GSK compound, which Silke is going to mention as part of her talk. So we're really lucky in the calcium signaling field and the cancer field to have really unique ways to basically modulate the activity of these channels, which other cancer researchers often don't have the luxury of. So if we just look now to, it would take too long to mention all the calcium channels that have been linked to different kinds of cancers. But if we just look at the TRP channels, almost all of them have been linked in some way to some aspect of calcium, but some really cut across different cancer types. And one of those is the ion channel TRPV6, which belongs to the TRPV family. So prior to our work, TRPV had been actually identified as overexpressed in prostate cancer. In fact, the first identification of TRPV6 actually was in the context of prostate cancer research because it is so overexpressed in prostate cancer. And we look at breast cancer cell, and this is our work now in breast cancer. When we looked at breast cancer cell lines, we found non-malignant cell lines generally had very low levels of TRPV6, but then there were some breast cancer cell lines that really had high levels of this ion channel. And indeed, this ion channels actually have increase in gene copy number, much like an oncogene like HER2 might. And then we look at these clinical samples here, this data, each one of these dots is an individual patient sample, and we look at levels of TRPV6. We can now see that basal molecular subtype that has the poor prognosis and correlates with triple negative actually has significantly higher levels than the luminal A and luminal B subtypes of breast cancer. So already within the breast cancer subtypes, there are some calcium channels which have different expression between the subtypes. And then when we look at what that consequence might be, here is again, our work looking with collaborators on how does the level of TRPV6 influence survival. And you can see here that if you're a woman with high levels of TRPV6, you're much more likely to die of the disease earlier than someone who doesn't have a high level of TRPV6 in their primary tumor. And we and others have shown, particularly groups studying prostate cancer, that TRPV6 is a regulator of cancer cell proliferation and growth. And so generally, collectively, when we see an overexpressed calcium channel in cancer, a variety of groups have shown that when you inhibit that calcium channel, you can actually inhibit proliferation or invasion or various other aspects that might be involved in cancer progression. And likewise, a variety of groups, including us, have proposed that maybe there might be some calcium channels that when they're overexpressed to a really high degree, that when we actually activate pharmacologically those channels, we might produce enough calcium influx to produce cell death. And we and others have now observed this. So this is our oncogene paper from 2017, which showed that there are some breast cancer subtypes that express TRPV4 to such a high level that when you activate them, you're actually able to induce a process called oncosis, which is a cell death pathway, or apoptosis in those breast cancer cells that overexpress that TRPV4 channel. So what I'd like to talk now is just talk about tumor progression. So hopefully I've convinced you that the calcium signal is really important aspects that are needed for tumor progression. Things like proliferation, tumor cell death, and metastases or motility are all regulated by the calcium signal. And there are some ion channels that are amplified, just like oncogenes can be in some types of cancers. There's also work being done by others showing that, that calcium channels are also really important in aspects of angiogenesis. So the ability to form blood vessels that will support tumor growth, and particularly TRPV4. What's emerged over probably the last 5 or maybe even 10 years is the fact that the calcium signaling probably is really important in the tumor microenvironment. And we and others have done work to show that environmental factors like the hypoxic tumor environment or growth factors, calcium signaling is really important in the way those things like hypoxia and growth factor have their effects on cancer cell progression and tumor progression. We and others have also shown that the calcium signal is critical in a process called epithelial to mesenchymal transition, or EMT. And that's where the cell adapts a more plastic stem cell-like subtype, and then that's believed to progress to resistance to therapy and also metastases. And of course, this group would know calcium is really important in many functions in immune cells, which places it as a key player in the tumor microenvironment pathway. And so when we think about the tumor environment, you probably would appreciate that when you have a cancer cell, not all cancer cells are the same. They'll have different phenotypes. They might even carry different mutations. But that's nothing compared to the variety of cells that surround and support that tumor growth. So we have things like cancer-activated fibroblasts, which can actually contribute to tumor progression. We have adipocytes, which have been shown to communicate with cancer cells. We, of course, have immune cells. So all of these things are functioning to form what we see as a tumor. It's not just the cancer cell. When we think about this, calcium signaling might be really important in how that microenvironment functions to support tumor growth and eventually metastases. So we're particularly in today's talk and in our lab recently have been interested in breast cancer brain metastases. So you probably know that breast cancer, along with prostate cancer, is one of the most common cancers. And some of them, particularly in Australia, are really well treated. In some economies, we're able to basically capture breast cancer early and treat them. But unfortunately, prognosis drastically reduced once a breast cancer cell spreads. And the kind of sites that a breast cancer will spread to is the bone, the lung, the liver, and the brain. And even though the brain is, a fairly rare metastatic event at around 10 to 30%, its consequences are quite drastic. So here we'll see survival over time for different kinds of metastases. So if you're a woman whose cancer has metastasized, obviously the outcome is going to be poorer than a non-metastatic pathway. But you can see here, if it's metastasized to the central nervous system, this prognosis is much poorer. So this is a critical area that we need to address. And we also need to address it because clinicians predict that as we get better at treating primary tumors and other metastatic sites, more women will eventually develop brain metastases. So it's expected that that proportion will increase over time as we get better treating breast cancer at the primary site and other metastatic sites. And when we step back and think, what does it mean for a breast cancer cell to go from the breast to the brain? It's really an amazing transition in terms of the microenvironment. So this is from a review from our collaborators, Joni Sanis and Sunil Lakhani, who are experts in breast cancer brain metastases. And from their review, they're really trying to define this event. So you can see here, we eventually get a primary tumor in the breast, and then there's a metastatic spread. First of all, the breast cancer cells, like all metastases, need to survive the bloodstream, but they then need to be able to enter the central nervous system and establish a new colony, which will then grow to an amount and survive that will have a clinical impact. And so when we think about it, the cells of the breast are fibroblasts, adipocytes, totally different to the cells of the brain. And yet this cancer cell has to work out how to survive in that environment. So it's really going from these two extremes. And so we look closely at that microenvironment of when a cancer cell spreads to the brain, then there's totally different interactions occurring, like particularly with astrocytes, but also neurons as well, and all cells of the brain microenvironment. And so we know from other researchers that intersect between cancer and the microenvironment of the brain is really important for the cancer cell being able to proliferate, become resistant to therapy, and also evade the cell death pathways that are trying to stop these cancer cells from growing. And also, interestingly, cancer cells can actually communicate with the brain microenvironment and actually do things like activate astrocytes. And the activation of those astrocytes is able to further feed back and promote metastatic outgrowth, or the actual progression of that tumor in the brain. And we now know through other researchers that a variety of signaling pathways is critical in both how the cancer cells communicate and vice versa with those astrocytes. But when we step back and think about those pathways, you can see that proliferation, evasion, cell death, metastases are all potentially also regulated by the calcium signal. And that's what we've been interested in. What is the role of calcium signaling in metastatic progression when it comes to the brain microenvironment? And so when we think about breast cancer metastases, there haven't been much studies, but there's some clues that it may occur. And so this is a Nature paper from Douglas Hanahan, one of the authors of that classic Hallmarks in Cancer review. And you can see here, this is published in 2019. And they showed some very elegant work showing that when you look at breast cancer brain metastases cells, so cancer cells in the brain that derive originally from the breast, they're able to form these synaptic-like interactions with neurons. And so they postulated that there were probably communication that might promote and allow breast cancer cells to survive and flourish in the brain. Not surprisingly, because of technical challenge, there's really been no studies looking at calcium signaling to see whether breast cancer and neural cells might communicate in some way and whether that might involve the calcium signal. And so we've been very interested in the idea of trying to work out, can human neural cells communicate to breast cancer cells, and can also cancer cells communicate to neural cells, to see how important those pathways might be in metastatic progression in the brain. So what's needed for us to better understand calcium signaling between breast cells and neural cells when it comes to breast cancer metastases? Well, first of all, we need some sort of neuroenvironment model that we think is robust enough to us to study that potential interaction between breast cancer cells and neural cells. We also need to measure calcium differentially in these two populations simultaneously. And because we envisage quite complex interactions, we need a high throughput way and a robust way to measure this interaction and assess this interaction. And so we felt high content live cell imaging was a way to do that. So there were the three things that had to be addressed for us to try and study whether calcium signaling may occur between breast cancer cells and neural cells. And so when we just think about the neuroenvironmental model here, we need to model that. So we could go a multicellular pathway, which is ideal, and that's what we want, because we don't just want astrocytes, we want neurons as well. So for that, we could consider a brain slice kind of pathway, which some people have done, not in calcium, but in other studies of breast cancer brain metastases. We also, though, want something suited to single cell calcium studies. So in that case, we might consider a primary cell culture. But again, that wouldn't be high throughput, and it also wouldn't be human. So really, we want a human model that is multicellular, suited to single cell analysis, but is also potentially high throughput. And for that, human neural progenitor cells proposed a possible model for us to start to look at that, trying to develop some kind of model to study this interaction between breast cancer cells and astrocytes neurons in human cells. So that was the task that we gave Silke for her PhD study, and she went on from there. And so I'm not going to actually pass on to Silke now, who's going to present her work on what she found in a PhD, trying to address and come up with a model that might be robust enough for us to study. So Silke, over to you.
Greg,
yep.
before we pass it over, would you like to entertain any
Sure,
questions?
if there's any questions about the model or calcium ceiling generally, yep, absolutely.
Yeah, so I'm not sure exactly how it's done. I missed a lot of these, but just unmute your microphone if you have a question right now. If not, we'll move on to Silke's presentation. Okay, let's move on. Silke?
Thank you. Okay, see if I can get this right. Yep. Perfect. Are you able to see that slide?
Yes. Mm-hmm.
Perfect. And I will just hide this one. Perfect. All right. So Greg left us off at the point of human neural progenitor cells. And if you've not come across this particular cell type before, it's a type of cell that begins in a proliferative state and can be induced to undergo a differentiation process to become mature neural matrices consisting of both astrocytes and neurons. Fortunately for us, there exists a commercially available immortalized version of this cell line known as Ren-cell VM. So we got our hands on some Ren-cell VM, and our first question with this model was whether we were able to use it to recreate the microenvironment of the brain that we considered was adequate. So here is an example of what these cells look like in culture when they're in their proliferative state. And they maintain what's often referred to as a polygonal, immature neural cell type morphology. What we found when we initiated differentiation in this cell model was that pretty quickly we saw a rapid change in cellular morphology into an interconnected network type structure, which was really encouraging because this is the type of morphology that we were expecting. But this didn't give us an idea of what exactly we had in our cultures in terms of cell types. So we took those cultures and we stained them for a marker of astrocytic cells, GFAP, which you can see in red, and neuronal cells, which you can see in green. And what we found was that while the proliferative cultures had largely no or modest levels of expression of these markers, following differentiation, we saw increased expression of both of these markers throughout the culture. And really encouragingly, expression of these markers corresponded with cellular morphologies that we would expect. In particular, these beautiful elongated cell bodies that we were seeing in the cells that were staining quite positively for the neuronal marker. If we then took these matrices and we ran them through an automated cell type detection software, we found that the matrices were consisting of approximately 70% astrocytes and approximately 8% neurons. And this was reasonably encouraging because this ratio of cell proportions was representative of the region of the brain that this particular cell line was originally derived from. We also wanted to check if this expression was maintained over time. And what we found when we looked at the neuronal marker, and here designated as MAP2, as well as the astrocytic marker GFAP, was that we had significantly higher expression after just 4 days of differentiation, and that this expression was maintained until at least 14 days of differentiation. So collectively, we had what we thought was a robust model of the microenvironment of the brain, but that didn't answer whether we were able to use this model for studies with breast cancer. So here is an example of some of our first work attempting to co-culture our neural matrix. So these a differentiated neural matrix that you can see down the bottom here with the breast cancer cell line MDA and B468. We gravitated towards this particular breast cancer cell line because it's part of the triple negative breast cancer subtype that Greg was telling us about earlier. And in addition to being particularly difficult to treat, this particular subtype is the second most common subtype to metastasize to the brain, which is one of the reasons we gravitated towards this area. And what we found was that when we co-cultured our breast cancer cells with the neural matrix, not only did the breast cancer cells survive in this microenvironment, but they were able to proliferate over time. So collectively, these results gave us the first element of the project that we required, a neural environment and a successful breast cancer co-culture model. The next section that we required was a means to measure calcium. So I would probably talk to this slide a little bit longer for any other audience, but I think for you, suffice to say, is that we stably expressed JR-CAMP in our neural progenitor cells, and we did a number of experiments to determine whether expression of this sensor affected the cell line's ability to differentiate, as well as confirming that it didn't affect the proportion of cells that we were seeing in our differentiated matrices. So were we still getting the same number of neurons? And we were. And we stably expressed GCAMP6 in our breast cancer cells. And now that we had cells that we were able to measure calcium in, we were able to go back to the neural progenitor model and start to ask questions like, did it have calcium signaling activities like we would expect for this particular cell type? So this is an example of what we were seeing when we had these cells in culture, just in monoculture. And although it doesn't appear like it, both of these videos on the left and the right are actually playing at the same time. So what we found was that while the Ren cell in their proliferative state maintained a largely quiescent calcium activity, following differentiation, this particular cell line developed spontaneous calcium oscillations, which was really encouraging because it's what we would expect based on previous reports. So once we had this particular model set up in a way that we were happy with, we then turned our attention to high content live cell assays using the model. And so this video that I have for you here is an example of our co-culture where we're just observing basal calcium signaling, and this particular movie just plays on repeat. And what we found with our co-culture model was when we just observed basal calcium signaling, the differentiated Ren cell matrices maintained that spontaneous calcium activity that we were observing in these neural cultures in monoculture. We were also seeing spontaneous calcium activity in some of the breast cancer cells as well. And we had actually previously reported with this particular cell line that spontaneous calcium activity was a feature. So this suggested to us that that the cells, while they were in co-culture, weren't dramatically changing their basal calcium signaling. But it didn't tell us everything that we needed to know. We were really interested in the potential of the communication of a calcium signal. So to be able to assess the potential of calcium communication in this model, what we required was a means to selectively induce calcium influx into one of the cells in the co-culture. So if I ask you to think back to those influx pathways that Greg so kindly set up for me previously, one influx pathway that we particularly were interested in very quickly was that of TRPV4. And that's partly because, as Greg alluded to previously, we had noted that some breast cancer cell lines highly expressed this particular channel. And the breast cancer cell line we were using in this model was one such example of this. And what we found, really fortunately for us, was that the Ren cell neural matrix model actually entirely lost expression of this particular channel as a result of differentiation. And as Greg has already, and as Greg also mentioned, we had a highly selective activator for this channel in our toolkit. So our next question was, what would happen if we took our co-culture and selectively stimulated calcium influx in the breast cancer cells alone? And so this is another video that's just going to play on loop. And the time point of agonist addition is going to be noted at the top left here by its name appearing in the video. And so what we found when we added our activator into our co-culture was a couple of things. Firstly, we were finding that the breast cancer cells responded with increased calcium levels, which is exactly what we were expecting. But we also noticed that what we thought we were seeing was increased calcium activity also in the neural cells. And I've got some areas here that I've highlighted that we thought were particularly interesting. And there were a couple of elements about this particular activity that we thought we could see by eye. In addition to the increased activity, we thought we were seeing potentially a period of delay between the time point of an addition and the commencement of calcium activity in the neural cells. We also thought we were potentially seeing increased calcium activity specifically in neural cells that were in closer proximity to the breast cancer cells in comparison to regions of the matrix where the concentration of breast cancer cells was relatively sparse. So just looking at the data, looking at those movies was unfortunately not enough. We then needed to really try to tackle the analysis of this information. So when we started to break it down with our analysis, our image segmentation, what we found was what we could see by eye, thankfully, namely that addition of the activator increased calcium levels in the breast cancer cells and corresponded with significantly higher peak calcium levels in the treated group in comparison to the DMSO control. We also found that peak calcium levels in the neural cells in co-culture with breast cancer cells had significantly higher peak calcium levels following addition of the TRPV4 activator, and that this difference in calcium levels was completely absent in our neural cells maintained in co-culture and treated with the same activator. And so collectively, these results suggested to us the potential of the propagation of the calcium signal from the breast cancer cells to the neural matrix. But if we think a little bit further about that data we were seeing in the video, one of the things that we were losing when we were looking at it at this population level was that we couldn't really capture the fact that what we saw by eye was potentially a period of delay and not all neural cells responding. So in order to try and decipher our information a little bit better, what we really wanted to try and define was whether we could tease out from our data the neural cells that were responding that were in closer proximity to the breast cancer cells in comparison to the neural cells that were further away. And so to address this, we developed a custom segmentation and data analysis approach where we determined spatial information for every single cell within our imaging field, and then we're able to separate our neural cells within the field into two groups, either proximate or distant, as we've defined here. And we did this to begin with arbitrarily based on a value that we had defined. So in this case, we see it here as D, which we defined as the diameter of a breast cancer cell. So neural cells that were less than this distance away were considered proximate, and neural cells that were further than that distance were considered distant. Further than that distance were considered distant. And even just binarizing the information in this way started to reveal some fairly interesting trends. So the data that I have on this slide all represents neural cells in co-culture. And what we could see when we were looking at the neural cells distant to breast cancer, but still in that treatment group, was that we were largely not capturing much calcium activity when we were pulling random traces out of the data. And if we were to consider oscillations over time, we were finding that the number of oscillations were fairly consistent through the imaging time. And if we compared that to our neural cells that we classified as proximate to a breast cancer cell, what we found was pretty stark differences in that calcium activity, both in those random traces that we were pulling out, as well as total oscillations over time. And in particular, what we could really see in this data was that period of delay that we were observing we thought we were observing in the videos, which was consistent with the idea of the propagation of a calcium signal from the breast cancer to the brain. Quite satisfyingly, when we looked at peak calcium levels within the 10 nanomolar treatment group of neural cells that were either distant or proximate to a breast cancer cell, we actually found that cells that were proximate had significantly higher peak calcium levels in comparison to neural cells that were distant. One of the disadvantages of looking at the data in this biorized way, however, was that we were losing some of the nuanced information that we could really have gathered from the fact that our data was actually continuous. So we decided to look at the data in one more way that wasn't binarizing it into those two groups. And so here we have an example of how we decided to approach that. On this graph here, every data point represents an individual neural cell in co-culture with breast cancer cells, with the cells stratified by their minimum distance to the closest cancer neighbor. On the y-axis, we have the peak calcium level for that individual cell for the entire imaging time. And our colored graph represents the number of oscillations that we detected within each cell during our imaging period. And what we found was that while we had the neural cells in co-culture with breast cancer in our DMSO control treatment, we couldn't see much of a relationship between calcium activity and distance to a breast cancer cell. When we selectively activated calcium influx in the breast cancer cell, we started to see a clear relationship between calcium activity, both in terms of peak calcium levels and oscillations, and minimum distance to its nearest cancer neighbor. And so collectively, what we took from these results was that the neural progenitor model that we used provided us with a robust multicellular model suitable for assessing calcium signaling at a single cell level, and that activation of calcium influx in breast cancer cells is able to produce cytosolic calcium changes in neural cells. These cytosolic changes are related to the proximity of the neural cells to the breast cancer cells that are responding to the TRPV4 activator. And collectively, we think that these results suggest that disrupting calcium signaling in the microenvironment of breast cancer metastases sites may represent new opportunities for therapeutic intervention. So to wrap up, I'll just say contribution and thanks to the people that were involved in this work, including members of our lab and our collaborators that Greg mentioned before, as well as to acknowledge our funding sources. And also thank you to all of you for listening to our talk. I will stop presenting.
Thank you. Thank you. That was great. I'm going to take the privilege of asking the first question. I'll go back to your talk, Greg. You showed some data. I think these were TRPV4 overexpressing cells, and you showed what happened when you activated TRPV4 pharmacologically, and you put them into cell death. Or what happens if you? I'm wondering about whether calcium, how plastic the calcium channel, calcium signaling repertoire is. So if you did the experiment, the converse experiment, which I think would be the therapeutic one, which would be to block those channels, can they upregulate some other pathway to compensate for that?
So I think for the genes, the calcium channels that are gene copy number increased, I think there's less less opportunity to compensate for. So things like TRPV6 that have and TRPV4 that have an increase in copy number, then if you like, that gives the cancer cell that property for long term, so it's hard to compensate. TRPV4 is a really interesting channel because it's also involved in EMT, but some aspects of EMT. So we've shown that if you inhibit TRPV4, you can actually stop the metastases. So we know that eventually, well, I would probably say this, we don't know yet clinically if long-term inhibition of calcium channels over a long period of treatment will activate a different kind of resistant pathway. Because if we'd normal therapies, we'd get upregulation of peak glycoprotein and MDR-HPAs and things that trump drugs out. And we also know cancer cells are very good at compensating. There haven't been enough work to know over a long period of time that would happen. Now, my personal bias is that maybe aura channel, which we're very interested in as a therapeutic, we don't see increases in gene copy number. I can imagine cancer cells getting around that pathway. For TRPV4 and TRPV6, where there's increase in gene copy number, it might be like HER2, and it's just a bit harder for cancer cells to overcome resistance. But we have no clinical data yet to indicate that's the case. But my feeling is it will happen probably for calcium channels that don't have an increase in gene copy number, that the cancer cell will probably upregulate another pathway, another calcium channel that might perform that similar function. Yeah.
Just unmute yourself if you have a question.
Yes, I have actually two questions, one for each. The first question is actually for Silke. Any idea of how the breast cancer cells actually communicate with the neurons beyond just an increase in calcium? And the second question is kind of related a little bit to Kevin's questions. If you have two different channels in the same cells, do they have the exact same role, or each one of the channels provide calcium that will do something else, like proliferation, metastases?
I'll do the second one, Silke, you do the first one.
Sounds good. So yeah, this is a question that we're very interested in. We have a suspicion about what we think is happening, but we haven't been able to prove it experimentally yet. So when we were first looking at the data with the propagation and noticing that there was a distance related element to it, we initially started to assume that it was potentially direct mechanisms. So something like potentially a gap junction that was shunting calcium across from the breast cancer into the brain. And there's some evidence that this is potentially possible that had been done in a neural model with melanoma, but it wasn't demonstrated with live calcium, so it wasn't really clear if that was actually happening. But when we then started to look at the data at that continuous model, we had cells that were responding that were further away than you would expect that direct gap junction to be. So it's actually led us to suspect that there's potentially a second mechanism at play, potentially the release of soluble factors like ATP. We'd previously done work that suggested that when you scratch 468 cells, they produce calcium waves across a monolayer, and that this wave was as a result of both direct contact and release of soluble factors. So one thing that we are quite interested in looking at are things like what purinergic receptors are expressed in our neural cells, and if we were to add in inhibitors of those purinergic receptors, do we see a reduction in this communication, for example?
And so my answer to do calcium channels do different things? I think the best model we have in breast cancer is our people's studies of epithelial to mesenchymal transition, which is where, and 468 cells, the cell line Silke uses is the classic example there, where it's a very plastic cell. So it has a property of epithelial cells in that you probably saw they're very rounded, but yet they are triple negative. And what you can do is you can treat them with things like EGF and convert them into what's called a more mesenchymal metastatic phenotype. Now, a whole different suite of calcium channels can regulate different aspects of that EMT transition. So TRPM7, for example, is a really strong regulator of vimentin expression, which is a marker, whereas TRPV4 is a regulator of a different suite of genes associated with that. So in that model already, there are different calcium channels that particularly regulate the different genes that are activated by the same growth factor in the same cell line. So, and that's why I think targeting EMT, getting back to Kevin's question, might be quite challenging, because that's an area where if you try and target that transition, there are other channels which probably might be able to contribute to that transition, even if you knocked one out. But that's an example where different channels can do different things in terms of a pathway in cancer. They can't exactly move one in for the other.
Thanks.
Hi, this is Don Gill. Very nice talks, Greg and Silke. Really enjoyed it. I like the whole concept of thinking about how cancer cells, for when they metastasize, how their environment, that's a really interesting sort of concept, and one where calcium signaling, it's a nice handle that potentially could have. So I just had one simple question. In the first, that's really for both of you, but Silke showed that when you first looked at the cells, the Ren cells, and the co-cultures, and I think you were looking at calcium signals, there was a movie. One side looked like it was completely static, and the other side with the co-culture, I think, you could see many oscillations of calcium. So that's quite remarkable that the Ren cells were sort of static, but I didn't understand why some seem to have very high calcium, some very low, and they maintained that, or were those fluorescent signals, were they calcium in those pictures, in those movies?
Yeah, so teasing out what was happening in just the Ren cell model, in its calcium signaling, both in our brain metastases model, but also as in the model itself, is actually something that we've been reasonably interested in. One of the other elements of the project that I was doing as part of my PhD was looking at why does it seem as though what elements of remodeling is involved in the Ren cell as a consequence of that differentiation. As you say, it starts in that quiescent state, and then it moves to a more calcium activity, higher levels, but some of the cells appear to have high basal calcium activity. And so this is one of the advantages that we've had with our really nice advanced single cell data that we've been developing as part of the co-culture model. We also did some work trying to tease back, are the responses that we're seeing in the calcium data related to particular cell types. So one of the disappointing things about just looking at the calcium is that we can't see if the cell that's responding is an astrocyte or a neuron, because we know it could be one or the other. So we've been doing a lot of work teasing together staining data and calcium data, and then using automated image detection to piece those two together, and then to grab that data and tease it out a little bit further. So this is something that I think we're still in the cusp of truly understanding in the model, but it is something that we're quite interested in. Greg, did that?
Yeah, and also we're limited. I mean, we're lucky in that we've got this technology and we've got these instruments, but we still need to use genetic encoded sensors, which are very difficult. Like, you could have a cell that just expresses more of the sensor, and so some of those cells that might look quite bright, they might just be expressing more. But we need to do that, because if you do a small molecule stain both the cancer cells and the neurons, and because it'd be hard to tease out. So even though once we did the analysis that Silke showed after the movie is all relative, delta F on F, those movies are probably influenced in part in terms of the basal brightness could be related to the morphology of neuron being more rounded. It might appear brighter at the start, but that's why Silke's now, it might be a different paper, but she has developed a technique where after the calcium assay, you can take it out, stain for neuron markers, astrocyte markers, and then define those differences in terms of calcium. My gut feeling is Silke, it's probably the astrocyte being the predominant population. It probably is astrocytes, maybe neurons, but my gut feeling is it can't be just neurons, because you wouldn't see that many cells change in terms of the location. And the delay is interesting too. Like this group, this delay is interesting to us, whether we thought maybe it's exosomes. Like, it's very unusual for us to see that very consistent delay between activating the cancer cells and then stimulating the neurons. That's an element that really we see, but we're really interested in exploring.
Yeah, very, very interesting. Really interesting approach and interesting results. Thank you. Thanks.
Jeremy, you had your hand up first, I think.
Oh, sure. Thank you. The question about the co-culture and the calcium signaling, I'm wondering if you have any evidence for communication back the other way, so from the neuronal cells back to the cancer cells. So I don't know if there's a selective agonist, thinking like glutamate maybe, that you could put on that.
Silke is definitely the best person to talk about this.
Okay.
So the reason that Greg and I are both laughing is that I think this is the
Painful
white
question.
male that I chased through my whole PhD, because every activator that we tried that would successfully activate one cell that wasn't the TRPV4 activator would also selectively would also activate the other cell. So we eventually reached the stage where we think it's very possible that there's a bidirectionality to this this signaling, but we think that in order to test that, we're going to need to be doing some genetic modification. So either knocking in a receptor to the red cell model or knocking out something in the breast cancer line.
What I think we learned is that cancer cells express a lot of receptors, and they are really upregulated multiple calcium signaling pathways. So anything that activated a REN cell also activated the cancer cells. So I think it relates to Sean's question, which is a lot of things upregulate some calcium channels, and that's why gene copy number is important. But things like histamine and things you might not expect to give a response in a cancer cell robustly increase calcium in monoculture, so it wasn't appropriate for the co-culture.
Could you tease that out of your analysis in any way by doing some kind of temporal correlation, spatial and temporal correlation of the calcium signals to see if there's,
So you did a general agonist or not?
I'm thinking more just maybe even the spontaneous activity.
Yeah, so this is something that with the level of analysis that we could do from, say, a statistical perspective, we were feeling a little bit constrained. So after my PhD wrapped up, we actually started to engage another group at the university that specialize in calcium signaling and mathematical modeling. And so this is exactly one of the questions that we've asked them. Can they actually find a relationship between, because sometimes by eye it appeared as though we could potentially see this occurring, but it wasn't at a rate that was high enough that anything that we were doing to the data was really able to reflect that. And that really nuanced single cell analysis of the information was a little bit above my level of expertise, and it's exactly what they're excellent at. So potentially watch this space, because it's definitely something we'd be interested to know.
Okay, Antonio, I think you had your hand up next. For me anyway, you're breaking up a lot. It's hard to hear. Can the rest of you hear him?
No.
Try again.
Yeah, Antonio, can you maybe type your question in the chat box and Kevin can read it out loud after Anna asks a question? Great, thank you.
Yeah, thanks. Yeah, Anna, you want to go? You're up next.
Yes, thank you. Great talks. Thank you very much. Coming back to this communication, which I find fascinating, I was just wondering between the neuron cells and the breast cancer cells, whether you had any evidence about direct communication through membrane protrusions. So, for example, in primary brain cancer cells, these cells communicate by tumor microtubes. They call them different things, but actually what we see is intercellular calcium waves from, and that also is from neurons to the brain cancer cells. So I was wondering whether you had any evidence in your system.
I think, unfortunately, I think the short answer is not exactly. It's definitely one of the pathways that we explored while we were discussing the potential of different mechanisms. We didn't come across a clever way to look at it directly. I think Greg was one of our conclusions for that one, unfortunately. But it's absolutely something that we're not ready to rule out as a possibility.
For example, if you use an anti-connexin, drug, then you should be able to. I guess the resolution, the videos you showed, they don't have enough resolution to see.
Yeah, exactly. Yeah, we can't see it.
Perhaps, trying that'd be really fascinating. Thank you.
Antonio, I don't see your question yet. Maybe you're still typing. So while you're doing that, I thought I would ask one about. I was going to ask you why you're calling these things neural cells, but I guess you're calling them neural cells because they're loaded with the same indicator, so you can't differentiate the two. So you've already indicated that. But I wonder, and I appreciate the fact that having a co-culture makes more sense, but in trying to parse out who the responders are and how the communication works, I wonder if it wouldn't be better to use a single population, just a neuronal population or an astrocytic population. And just along these lines, is there an advantage to using these neuroprogenitor cells rather than some type of IPS cell system? That's more of a general question.
So Silke, do you want to have a go? I think in terms of monoculture, I guess we were just attracted to that idea of seeing two populations at once. And I guess if Silke's, we're hoping the staining of the different subtypes will work, but I guess that is a good suggestion that our gut feeling that it's astrocytes, we could just go to an astrocyte model and confirm that happens. So that would be something, Silke, that we could easily do if we don't, well, not easily, but if we don't actually have a robust system to actually post-identify what populations are responding, to actually go in monoculture and then confirm that it is able to communicate through astrocytes. And it also might make the image analysis, and we might be able to find new things, because that would be probably a more consistent way. We could probably do a clonal Silke and sort of get, even maybe use a FURA2 model and then have genetic encoded calcium, and then probably do some new things and more nuances in a different way. And Silke, you're the expert on the stem cell side. I guess we jumped at Ren cell before you got to the project, but are you able to comment on other models we could have looked at?
Yeah, so I think one of the, in addition to what Greg was referring to about why we were drawn the neural progenitor, even though it has thrown up some challenges for us, is that a lot of the work when we came into to this space was really looking at only animal models, and there wasn't really any nice cell culture that was human to human connections. So one of the real advantages that we had in this particular cell line was that it was that human origin. There are a few other progenitor-like models that I think we had, which would have been a possibility. And I think that our collaborators that we referred to, Jody and Sunil, who are those brain metastases experts, that have gone down that route. But one of the reasons that we had been drawn to, I think, as well as the Ren cell model in particular, is that it was one of the few in that space that that we had direct access to and experience with those collaborators, and there was some calcium data that had been published in it as well, although not a lot.
Oh, so a question in the chat box is brain organoids. Yeah, that would be great. We would love a confocal high content imaging system, but that's a little bit above our budget at the moment. But I agree, that would be, because I think that would be like true connectivity and stuff. I think that's the next level to do that. I 100% agree, Anna, that would be our dream to eventually go down that direction.
Can you hear me?
Yeah.
Can you hear me now?
Antonio typed in a question. Oh yeah, I can hear you, Antonio, now.
Okay, my question is for Silke. I would like to know if there is any direct communication between the tumor cells and the neuronal cells via gap junction channels.
We think that direct calcium communication is definitely a possibility in this model. And one of the things that we have played around with is the possibility of gap junction inhibitors in addition to the purinergic inhibitors that I was referring to earlier. We have a slight challenge in positive controls with the gap junction inhibitors, just showing that the cells can communicate maybe in a monoculture via this mechanism, and that the inhibitors will actually block that before we attempt to look at it in the co-culture approach. But we think that in looking at the mechanism, it's definitely an element that we're not ruling out just yet. There have been some suggestions that some brain metastases can form gap junctions with the brain, and I think connexin 43 and 26 have both been suggested to be involved in that signaling with other signaling pathways before. So it was absolutely we were 100% sure that direct contact was probably our number one candidate, and that was only really when we started to do that more continuous analysis of the data that we started to even consider, is there a secondary mechanism at play?
And there is a nice paper that does human melanoma cells with mouse astrocytes. I think it's that model that does explore gap junction interaction. Is that the one, Silke, that is up there? That's sort of like, so I definitely think it's definitely a possibility, but just hasn't been done in that human model. But that delay thing is interesting to us. Like, what does, if it's gap junction, I guess we'd think it might be faster, but it could just be that if there's a shunting mechanism, you might need to absorb a bit of calcium before you get that spontaneous activity. So, anything is really on the table at the moment.
So direct transfer of calcium from the tumor cell to have high calcium concentration to the neuronal cells, it could be possible.
Yep. Yeah, definitely.
Okay. Thank you. Great.
I have another question. When a tumor, metastatic tumor, grows in the brain, does the brain suffer? In other words, are you losing cell types in the brain? And as you're developing your mixed culture, as the breast cancer cells were growing, was that at the expense at all of the other of the neural population?
I think clinically you get, with metastatic brain cancer, you do get cognitive function fairly early. So unfortunately, women often report those effects with very small metastatic brain tumors, and then because there's nothing much that can be done, it's a monitoring thing. So our collaborators actually done some nice PET scanning work on progression, and there are interesting things about breaking down of the blood-brain barrier, and particularly there's a lot of work, Silke, on pressure within the brain. That's why I'm very interested in like the force sensitive channels. There's a lot of evidence that in the brain microenvironment you can actually get quite a high pressure just from the concentration of those cancer cells. Your second question in terms of, Silke, you can probably answer that one.
Yeah, in terms of total cell number, I think that's quite interesting. I actually think we could almost tease that out of our data potentially from our cell number alone. What it appears happens when we look at the co-culture is not potentially that we're seeing any dramatic changes in the co-culture itself with the breast cancer cells while they're in them, but what we do find is that the breast cancer cells seem to preferentially develop those tight connections with the neural cells with this particular cell line that we're looking at. And I think something that we actually found that I hadn't shown that was a surprising unexpected consequence of working with the co-culture is that we tried a couple of other breast cancer cell lines to see what they would look like when we were keeping them in co-culture with this neural model. And while some of them form those tight connections like the 468 cells, another breast cancer cell line that we were looking at, which was a luminal B type, BT474, and it was a brain metastatic variety, actually tended to form islands around it where no cells were growing, and they do suggest that there's quite nice evidence in breast cancer bone metastases that the effect that the breast cancer has on the microenvironment when it's arriving at that microenvironment can either be osteo forming, so can form more bone, but more commonly is osteolytic, so it destroys that bone. But it's by no means a consistent process, and this just from playing around with different cell lines in this particular model, it definitely seems as though breast cancer cells are not uniform in the way that they're responding to that microenvironment, which we thought was quite interesting.
And that's why we had to go, because those tight ones, although they're really interesting, imaging wise, it wasn't possible. We need to, for single cell analysis, they need to spread out a little bit more. But those BT, the brain derived breast cancer cell lines that we wanted to use originally just seemed to go straight for it. So our theory would be is that because 468 cells are more that plastic type, they can be both epithelial and mesenchymal. They're probably plastic enough to get in there, interact with the neurons, and just have a symbiotic relationship. Whereas if you put those, I know that's my theory, Silke, if you put the brain-derived ones straight in there, they do exactly what say Kevin, which is they just seem to clump with each other, grow as masses, and push all the neurons away. Whereas we were trying to get a model where we could actually, they were more symbiotic and being supported. Whereas the other ones just, you can imagine that might be late-stage breast cancer where they just got those really high pressure points and it just pushing cells away.
There are several compliments in the chat box and a couple of suggestions, so you might want to look at that. I can't tell if anybody has their hand up or not, so just unmute yourself if you want to ask a question. Great. Well, I'll echo the sentiments. Really fascinating. Both talks are a great system too. Wow. Really cool. Thank you so much.
Our pleasure. Thank you. Thanks for having us.
Yeah, thanks everybody. That was super duper. Thanks, Kevin. And thanks
Happy
to
St. Paddy's Day, everyone.
Yeah.
Thanks a lot, Greg.
Brilliant.
Thanks. Great talks.
Take care, everyone. See you again next month. Take care all. Bye now.
Thank you.